Hi, team. Welcome to final prep. How to study for a test when you only have 3 days. I enjoy the analogies of the Friendship is Magic team, mostly because I have a friend who's an animator on that team, and also because I like to teach as if I'm teaching to children, even though I hate teaching children. So anyways, you'll see Rainbow Dash periodically because that's the thing I do. The organization of this is going to be, first we're going to talk about the anatomy of the test. Then we'll talk about last minute studying techniques. And then we'll talk about day of planning and structure. When talking about the test, we will actually go and look at the practice exam. And I'll talk a little bit about that and show you things and show you how I take exams. And then when we talk about last minute studying, we'll go over all of the topics from this course as put out by Dr. Fitzpatrick in her Word document. I kind of broke it down and made some of her emphasis louder. So that's about it for modifications. And I do want to emphasize, per usual, that I would probably get a B in this course. I'm knowledgeable about some things and not knowledgeable about other things, but I am in your corner and I'm here to be helpful. So with that caveat in mind, and I just realized that you asked a question, you got a sticker. Okay. So about the test, multiple choice, short answer, short questions and long questions. These are the 3 categories that she does. Multiple choice questions are like clicker questions. The way you study for them is by looking over the clicker questions. The short questions are like the lab quizzes. Sometimes there's a couple of them that refer back to each other. Sometimes there's like multiple choice short answer combined self-referential things. Long questions are like mini labs. There's a whole page of reading before you actually start to answer any of the questions. So we'll talk about how to approach these different styles of questions.
How long is going to be the exam overall?
I think it's 3 hours, 2 hours? 3 hours, I think. Yeah. Yeah, 3 hours. Okay. Okay, so multiple choice / true false, like quicker questions. Pay close attention to phrasing for these questions. This is one of the places where she's going to test your understanding, not just of the meaning of the words, but how they are used together, how they refer to each other. An example, mutant versus mutation, allele versus gene. So if we look at this, we just look at this complementation question here. When 2 mutants complement each other, they are alleles of the same gene. So complementation, that's not true, it's the opposite that's true. If they fail to complement, that means they're alleles of the same gene. However, there's a better cue that A is wrong, which is that she says when 2 mutants complement each other, which what does that even mean? It's two mutations that complement each other. So you know that's wrong already. And you are even more confident that it's wrong because there's the exact same option, but phrased better over here. When two recessive loss of function mutants complement, they're alleles of the same gene. That's the exact same thing, but said better, right? Which means that that's also wrong for sure. So you can rule those out, which leaves us with these three. Complementation has nothing to do with proteins. When two different genes affect the same phenotype, that's epistasis. And that leaves us with D, which is the correct answer. So this is how one does multiple choice questions. If you're unsure, don't spend too much time trying to figure it out. It's only worth a point. But also, don't leave it unanswered. If you by chance get it right, that's a free point. Rule things out that you can rule out. Like, I'm pretty sure that complementation has nothing to do with proteins. And you can rule out those two because they mean the same thing. It's just that one is said worse than the other. So both of them you can rule out. And then you can guess between the remaining options. If you're not sure if you can't remember the difference between epistasis and complementation, that's a 50-50 shot. Do it. Feel good? Mm-hmm. This is how multiple choice works. Now let's look at one more. Which of the following is not associated with P elements? Well, this is a chaotic one. I personally would skip it and come back later. Like for real, you go through the first pass and you only do the answers to the ones that like, oh yeah, I can do that. Flies are homozygous recessive for cinnabar, have blank eyes, and they lack blank pigments. Okay, so flies that are homozygous recessive for cinnabar means they don't make cinnabar, which means that they have cinnabar eyes. So they're orange. So we already know we're picking between A and B. And then they lack which pigment, pyridine or omochrome. Cinnabar has orange eyes and omochrome is the brown pigment. So they are orange eyes, which means that they don't make brown pigment, and omochrome is brown. So that's B. Pyridines are orange. Orange plus brown makes red. Yeah. Yeah. Yes. The natural wild type fly has red eyes. Yeah. Last time I checked. So does this make sense about prioritizing? And then when you come back, if you're working on something, and then they say 10 more minutes, stop what you're doing, go back and answer all the multiple choice you left. Right? Because you're gonna, statistically speaking, get more answers, get more points, circling essentially roughly at random on those questions than you will panicking while you're trying to read the description of an extended answer question.
This is not like filling the.
No, I just circle the damn thing. Does that make sense? Yeah. So at the 10-minute mark, stop, go back from the top. That's how you do multiple choice. Any questions about multiple choice strategy? Ooh, there's a chat.
Do you know the number of questions or marking breakdown?
I don't. I think it's out of 200. 200 or 100. But it's either 100 or 200. And my suspicion is because she had one of her example extended answer questions that was worth 50 points. Like one of that like mini lab thing. She's not going to make a single mini lab quiz question, the single mini lab question with like a bazillion different options. That's not going to be 50% of the exam. So I hypothesize that it's going to be 200 points, but that's a good thing. That means there's more points to get. It means that tiny mistakes mean less. What is the difficulty of the exam going to be like? It's going to be a little bit easier than this practice exam, but not much. Okay? And it'll be curved, I suspect. I'm not going to promise it, but I'm pretty sure this course is going to be curved. So be good to yourselves, be patient with yourselves, answer what you can. Okay. Any more questions about multiple choice? Let's move on to short answer. These are like the lab quizzes. Remember where there's like a few words here, maybe a paragraph here. And there are a few, put that over there. There are a few kind of codes that I want to emphasize. But first, the format of this question, there's a setup, right? So there's a little description. It's like a paragraph long thing you have to read. And at the end, it says total points. And that'll tell you like how many of the following things are related to that. And so if you start skimming that and you have no idea what it's talking about, skip it, come back later. Right? And then you know how much to skip to come back later. This is supposed to be not a full page, just a few lines, not a full page. But as you're reading that paragraph, highlight important information as you go, even if you're not going to read and answer it right away, because highlighting it or paraphrasing it in the corner will help you know, be prepared once more of the juices are flowing when you want to go back and read it. Take a few notes. The sub questions, there will be instructions. And again, highlight the things that matter, label or paraphrase this part. If it's saying like complicated, like describing a pedigree, draw the pedigree. Phrases like briefly describe mean write a paragraph, maybe draw a diagram, don't go crazy. Does that make sense? Briefly describe, don't write me an essay, just a paragraph. Explain means tell me how you do it, what are the steps? That's different from how, which means prove you know what this thing is and how it works. Talk to me like I'm a child. So, or sorry, not how, why. That's different from why, which means prove to me that you like spell it out. I need you to say the P factor is inserting itself into the gene, which means that there's a long stretch of DNA that's new and doesn't belong there, that's in the middle of a gene, which means that it's going to be transcribed into a long thing, which is more likely to disrupt protein function than it is to improve protein function. Like every single step, walk me through it. Sound good? Okay. Kathleen grades in half points, which is great. It means that there's a lot of opportunity for you to get points, but also it means that if you see a 1-point question, there are 2 aspects to it. And you should try to figure out what that aspect is, especially if it's a why question. And it's worth 1 point, or a why question that's worth 2 points. 2 points, write me a paragraph. Tell me why.
What if it asks like what gene did you use for this? Like, do you just write the gene name, or like
What?
like
If it just asks you a question where it's like I use this balancer, do you just say you use the balancer, or do you say I use this balancer because blah, 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 blah? If it just asks you, that's what you're asking?
yeah, like if it's a 1-word question.
It's a 1-word answer.
Oh, okay.
Yeah.
Okay.
Yeah.
Gotcha.
Don't write me an essay if it's not explicitly asked for.
Okay.
Yeah. And I'll go into more detail about we're not testing your writing ability here.
Question. If you don't know the exact wording for it, could you describe it in a couple of lines or something?
Yes.
Because I know that in the quizzes we have done that, and if she understands that you know the concept, but you don't exactly remember the wording, did give us some points.
Yes, absolutely. If you're not entirely sure what the magic word is, write a couple lines. Don't write me an essay. And if it's going to, if you've been writing and then erasing and then writing and erasing, circle the answer. Point arrows to it. Cool. All right. Ready for long questions? No, never. Think of these as mini labs. And there are several different stages to them. So do these last. Like on the first pass through, skim them for sure. Get a feel for what the topic is, but don't look at them in any great detail. You want to mine for easy points before you get to the hard points. Make sure that you've got as many of the easy points as you're going to get in a timely fashion, but things you don't know, leave them. And then you start with the long answer questions that you feel most confident in. And the way you start that is you skim the setup. And then you skim the questions so you know what information you're looking for in the setup. And then you read the setup closely.
When you say the setup, what do you mean, like skim the setup? Like,
what a great question. Here, let's take us to this thing, this monstrosity. Where are you? Is it this one? Where does this start? Longer questions. Here we go. Longer questions. So the setup is this whole thing. Starts question 16, and then there's a whole damn page of information. And then we have A at the bottom on the next page. This is the setup. And I've highlighted some things here that one should highlight as one reads so that they'll jump out at you. Bring a highlighter. Feelings. Okay, so you read the setup, you highlight important parts, draw diagrams or rephrase as you go for clarity. If something is confusing and then you finally understand it, write it down in the corner, in the margin. I can give you points for things you figured out, even if they're in the wrong place. So tell me the things. Show me your thinking. Step 4 is answer strategically. So answer the things you know first. Write notes for things you're less sure about, but don't spend too much time here. And for things you're less sure about, write bits you are sure about or suspicions. I'm pretty sure this is asking this thing. Or this is clearly requesting an equation that I haven't memorized. But these are the elements, because I might be able to give you partial credit for that. And then 5 is move on to the next one. If you aren't finished with one, but if to get more points, you're going to have to spend some real time processing, don't do that. Go to the next one. Read that one, get those points. And then those other points will be easier to get later while you've been thinking about it. You got 3 hours.
this is not enough.
I believe in you. Okay. Does that feel better? Does that feel slightly more doable, these question types? Okay.
Like to believe so?
You would like to believe so. Let's real quick look at this thing here. So here's, I just want to point out the way that this is written. This is a story, right? So you have some deletions on chromosome 3. You're interested in mapping them in terms of the genes that are deleted. That means we want to know what genes are deleted. That's the goal. You have some point mutations that are recessive lethal. That's good to know. The point mutations are these lines. Here's a map of what's known so far. The shaded bars, the chromosome reason, the lines is the the line, these long lines below it are the deletions, and the short lines show the part of the chromosome, each line shows the part of the chromosome that's been deleted. Here are the recessive lethal mutations in these little lines. There are a lot of genes here, but you've designed primers for only 3 of them. These are our 3 genes we're going to be working with.
So the primers are genes.
Genes. These are the names of genes. You've made primers for those genes. So you can check to see if those genes are present. And we have these deletions, right? Yeah. You're performing controls, you're running a normal PCR, and there's a fire alarm in the middle of it. So you hurry up and finish. That's cue for, oh, there's going to be mistakes, isn't there? And so then obviously the first question is, what mistakes have you done?
But then I think there's a follow-up saying that you shouldn't say the most obvious ones.
What?
Wasn't here. I read in one of the questions, I think it was one of the question papers, where it says that there's a fire alarm and you shouldn't go through the apart from the obvious mistake. So I forgot to put the primer or this, that.
Yeah.
Explain what else could have happened.
Really? Oh, it says a later question, what other reasons that could have been wrong? Oh, I see. That's hilarious. I mean, I love it. I love the emphasis on creativity. There are a lot of different things that could have gone wrong. This is why PCRs are terrible. Yeah. So that's how you read these things. And then you get a feel for these questions. And again, don't think about them linearly. Or think about them like those labs where you looked at all of the things and then you kind of worked as a group and figured out what the order is. But it's rare that you start at the top. Sometimes you don't even get the papers in any logical order. So you do want to read it all and then go back. Okay. Let's talk about how to study. Is this a logical order? Or do I want to talk about day of stuff? No, let's talk about how to study, and then we'll go back to day of stuff. How to take an exam. Okay, so study in ways that force you to combine different topics. This will help you learn them better. This will help you remember them if they have context, kind of fit into your world view. One of the ways that you can do that is to make a vocabulary list and then try to use those vocabulary in reference to each other. You can also make an association map. You can have all these words out and you can try to connect them in different ways. You can print them out on a printer and then cut them up and draw lines between them or whatever you want to do. Working with the words so they mean things and aren't intimidating, but have relationships with each other. Another good way to think about this is to make a list of the class topics, which Kathleen has helpfully done for you. And then you can make a billion Venn diagrams that compare different techniques or processes. Or you can make a flow chart, a way of connecting all the topics together into a single narrative. Try to imagine a world in which you did literally all of the things mentioned in this class to a united purpose. How would that look? Another thing to do is to work through the topics guide in the next section. If you're someone who you feel like you understand this material, but you need reminders, or if you're someone where you feel like your understanding is very random, like you know a lot of this very well and some of this you don't remember at all, then it's better to go through topic by topic. And the other thing is the practice exams. The practice exam that I had you do is one of the many. You go to modules. There we go. It just did it magically for me. I love that. So here are all the clicker questions. Ah, great. These are your multiple choice question studies. Do one an evening. Like just pick the topics you feel least confident on and go through them. Or, you know, do yourself a practice exam, print them all out, shuffle them up, and then run through them. Sit down for an hour. Make it happen. There's also old midterms. Some of these questions won't fit into the topics list that I gave you. If the question you're reading isn't in one of these listed topics, or if we didn't go over it over the class, you will not be tested on that. Don't worry. Previous classes went over different material. For these things, don't read the answer key until you've already done the thing. Just say no. Actually test yourself. Try to solve the problem. That thinking process of, I can solve this problem, I'm going to solve this problem. That's how you learn. Don't just read the answers and try to make them fit. Because then you're not going to be able to pull up those answers. It's going to feel like half the story is missing when you're actually there. So actually test yourself. And these things here, think of them as practice for the long answer questions, right? Read them through and think, okay, when questions like this are asked, how do I answer them? Does that make sense? These are good resources. Use them. Spend a solid few hours going through here and running questions so that it's not scary to be faced with this type of question. Study group. Studies show that people learn better in groups. If you don't have access to a study group, try to explain concepts out loud to your stuffed animal or your deeply bored roommate, or your roommate who has headphones on and is playing video games. Like, explain it to somebody else, even if they are not paying attention. You using your words to try to explain it will make you realize what parts you don't understand. And that will help. Okay, careening on to the next stage. Are you ready? This is a segue. I don't do segues. I have terrible transitions. It's horrible. Okay, topics. I'm going to give you an info dump. That you're going to have this document, so don't feel overwhelmed about this info dump. Think of it as I'm giving you the texture of things, and then you can go back over it in detail later. How do I use this massive info dump? Use it as a checklist when you're collecting study resources. Don't worry about practice exam questions that don't fit into this list. And bolded things are things I suspect will be covered in depth on this exam. All of these things will be vaguely mentioned, or most of them. But there are a few that I think will be covered either repeatedly or in an in-depth question. So make sure you understand them. Make sure they're familiar. And again, I have not seen this test. This is my predictions. The test is already written and submitted. So when Kathleen watches this, she won't be able to change the exam based on my predictions. These are independent things. So caveat, 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 caveat. Don't at me. Okay. General topics, first priority. Lecture slide terminology, bolded words, words at the end of lectures, in summary slides at the end of lectures. Just collect them into a list and then try to define them yourself and then Google them and make sure you're right. If you do nothing else to prepare for this test, do that. The other category is the 202 material. The review terms covered in the first week and posted online. Look at them. Watch a couple videos. Make sure that you are familiar with these concepts. That includes pedigree analysis, justifying pedigrees, probability, chi-squared testing. You will be required to do a chi-squared test.
So then you be given the formula and stuff and the charts. Or the-
You will be given the charts, but not the formulas. Yeah, learn those formulas. I hate it too, sorry. And let's do just a real quick here to look at,
Wait, can I ask the formulas for what?
the formulas for the chi-squared test.
What formula is that?
EO
Yeah, observed
over
expected.
expected.
Oh, that basic stuff?
Yeah,
Will we be given the probability formula?
no. We're not giving you any formulas. All we'll give you is the P is how to find the P value with your chi-squared value. Hmm?
Have to memorize the mapping formulas as well.
Yes, and you're gonna have to explain them and why they are what they are. You have to understand the mapping formula such that you could make it up from scratch if you had to.
Speak of the hospital information.
Or skip that one.
Yeah, maybe so.
No, I don't think so. I think
Every
that's only
question.
one question. I bet you that's going to be an aspect in 2 questions.
Mhm.
Anyways, what I'm trying to say is week 1, review materials. Mini catch-ups. This is what she's referring to. These mini catch-ups. Just run your eyes over this material and make sure it makes sense to you. It's an important thing. All right, moving on. Understand meiosis profoundly and deeply with all your soul. This will come back again and again and again. She'll ask you things like, how is this possible given meiosis? You know, in the context of reproductive mysteries and pedigrees and the corn kernel thing, tetrads. She's going to say what stage of meiosis over and over and over again. If you are sketchy on meiosis, you will lose so many points. This is one that's going to just come back over and over. So when you're studying and when you're going through the lab reviews, and specifically, think about it through the lens of meiosis. All right, second category, methods. P elements. This has been a big theme of this course. You need to understand how they're used, how they work, how to do a screen with them. Balancer chromosomes. You need to understand this stuff. No, don't do that. Basics of rescue and MRI experiments. And how PCR works. Keep in mind that this is going to be a big theme. You're going to have to come back to that over and over again at different levels. How to find what makes a good primer, how to troubleshoot, how to identify primers, that sort of thing. And she's going to ask you to design an experiment. I would put money down that she is going to ask you to design a simple genetic screen for dominant mutations. Because this was very specific in that thing. Given starting stocks, so she'll give you stocks and she'll say screen for dominant mutations and know when individual crosses happen versus en masse crosses and why. This is going to be a P factor screen for dominant mutations. This is very specific. I think this is going to be a short answer question or a long answer question. A short question or a long question. I also think that this was a little too specific. The behavior genetics, how screening and rescue, and how these methods are used to figure out when/where a gene is needed for a certain behavior. That practice question about the fly behavior, I think, is really good material to go over. Does that make sense? So let's talk about lab specific things as a category. This is your third priority review topic. I predict that there will be a long answer question that asks you to essentially design the DMAP experiment, but with a different goal and a different set of mutations and different stocks in mind. And then this is going to be a long answer question with a bazillion different stages. I predict that that's a thing. This was too much detail. I think this is going to be a short answer thing. I think she's going to ask a fungi question. She's going to make you map using fungi. So be prepared. But I think that's a short answer. That's only going to be, or maybe like a medium, like that's going to be a 10-point question, whereas this fly eye color thing, it's going to be like a 20-point question, maybe more. Human genetics. This is going to have less emphasis, I predict. But there will be at least a short answer, if not a long answer, that is akin to the family that walks on all fours experiment. That will force you to look at chromosome rearrangements. It'll force you to look at STRs and SNPs and RFLPs. And they'll do that thing where you're trying to see what segregates with the phenotype. I predict that will be a question on this thing. Polygenic traits. She's not going to give you those calculations for the polygenic trait thing. You need to memorize it. I would be surprised if it didn't make an appearance, but I don't think it's going to be super emphasized.
Equation 2n plus 1 equation?
Yeah, but then there's like complications to it.
Yeah.
That was a whole thing. Does that make sense? Epigenetics. There will
Madam,
be.
can I ask a question?
Yes, of course.
If I ran out tonight and got a tattoo of all the formulas, right? Would I have to cover that up before I take the exam?
Yes. Yes. And I would notice. And I would say something.
Well, I definitely wouldn't try and sneak it in. It's a tattoo, so you know, it's on my body there. I can't hide it.
Yeah, how can you, yeah.
Permanent marker.
That's cheating. You will get written up for that, for sure.
Well, I won't do it. Don't worry. I won't
But
do
I'm
it.
glad. Good. So, X activation versus dosage compensation will be a question. It will be a thing. She might even have one of these questions, like talking about ZW issues. So trying to make sense, trying to kind of make up a way to do X activation / dosage compensation stuff in a different model. And this is a neat module because it has a bunch of example questions in it. Yeah. So that's my predictions for what's on the exam. We'll see how right I am on the day of. Segue. Day of. Come with all the things. Work smart, not fast. Look, it's cute. Write clearly and succinctly, don't panic. And we're going to go through them all. Come prepared. Prioritize physiological needs above everything else. Bring hard candies to fuel your brain. Your brain runs on simple sugars, not complex sugars. Please don't bring anything with peanuts in it. Please, whatever you bring, food or water has to be well sealed. But I strongly emphasize that you bring something with sugar in it, so that in those moments when you're just like have a blank head, you've been thinking too much, eat some sugar, it'll help. Bring something to drink, not too much, you don't have to pee 5 times, but bring something to drink, because again, you need to keep the machine well oiled. Take care of your physiological needs. Also, I can't believe I forgot to put this in here. Be well rested. Studies show that students perform better on an exam if they have had 8 hours of sleep and not studied, versus students who have studied hard but only gotten 5 or less hours of sleep. If the options are sleep or studying, choose sleep. You will perform better. Science. Please sleep. This is, wait, is this an early class? This is an early exam, right? It's an early exam. Go to bed early, take a Benadryl. Seriously, take a drugstore sedative. Sleep. You will thank me.
Imagine you miss the exam.
Well, you have to actually lie down before, like early enough, like lie down at 9, right? Yeah. Can we use a blue pen instead of a black pen? No, I'm gonna say no. The reason for that is that this test is getting scanned into a computer, and then I only get to see the scans. So what?
The blue won't be scanned properly.
Yeah, so make sure that your writing is clear and dark. That's the best way to be sure that you will get the credit for the things you know. Bring the very best tools. Test all of your pens and pencils beforehand. Bring whiteout and test it the day before. Bring your favorite pens, the cutest eraser, the fanciest hard candy. Feeling fancy will make you more confident, which will help you think more clearly. So make sure that your tools that you have, they feel nice to you, that you're working with nice things. Bring objects of comfort, something that will make you smile while you're panicking. Something that is a sensorily pleasing thing, a pleasant texture. These little doses of oxytocin, like a little picture of your dog, will help you stay calm and focused.
Yes, right?
Yes, and we will have some for people to borrow, but yes, please bring a calculator. Forgot that. Let's add that.
One of the important ones.
What?
One of the important ones. I think you could survive without candy, but not calculating.
For sure, I think so. We'll have calculators to pass around. So yeah, if you don't bring a calculator, that's okay. But if you don't bring candy, you might stop braining after an hour and a half, and that'll be an issue.
Or some from the movie.
Okay, so this is your list. Make sure you get to check things off it. Do the things.
So the original,
Do not bring alcohol, Avash, Jesus Christ. Yes,
the paper has to be written in pen. Can we not write in pencil?
you can write in pencil.
Right?
Yes, absolutely. And I say dark pencils more than one, black pens more than one. The reason that you want to bring a pen is if you write in pencil and then you erase it and then you write in pencil again and then you erase it and then you write in pencil again, then you erase it, and now there's a spot on your paper that's permanently dark. You're going to want to wipe that out, and then you're going to write in pen on top of that. Does that make sense? Just as clear and tidy as possible. And again, that moment of panic, you want to be able to make that tidy. You want to be able to overcome that moment of panic with something crisp and clear. Work smart, not fast. I was talking about this earlier. You're going to do 4 passes on this test. The first pass, you're going to quickly answer all short multiple choice questions. Don't leave any blank. If you don't know the answer, rule out what you can and guess. You're also in that first pass going to skim all of the long answer questions and get a feel for them. Maybe highlight a couple things, write a couple things in the margins. Second pass, you're going to answer the short questions you feel familiar with. You're going to read the setups for those short questions carefully and highlight important details and words. You're going to draw out complicated word questions into a format that makes more sense. You're going to make sure you know what every point is for. Then you're also in that second pass going to skim the long answer questions and prioritize based on familiarity. Answer the easy parts only if it's obvious at this stage. Write short notes about more time-consuming parts. The third pass, some students are going to feel that time approaching. If you're feeling that time approaching, you get to make a judgment call. Either answer those long answer questions if there's one you feel super confident on, or go back through the short answer questions, make sure that you have gotten as many points as you can. But the third pass normally would be answer all the long answer questions, but in order of most confidence. And the final pass you do, no matter where you are in the long answer questions, 10 minutes before the end. 10 minutes before the end, you're going to stop and you're going to go back to the top and you're going to find all those short answer questions that you left because you had no idea what they were talking about. And you're going to do your best to answer them. Then you're going to check everything for completeness. Is every point accounted for? If you've got extra time, that's what you're doing. You're not just done. Don't leave early. Don't do it. I know you want to. Don't do it. Go from the top. Check your work. Make sure that you didn't miss anything. I don't know how many multiple choice, short answer, or long answer questions there are. I do know they're going to be fairly evenly distributed. So I would predict about a third of the questions would be short answer, a third would be multiple choice, and a third would be long answer. Write clearly and succinctly. These tests are being scanned into a computer. Write your answers in dark pencil if you want to use pencil. And like I always use pencil. Check your eraser. Make sure your eraser doesn't smudge. If it does smudge, use a whiteout and then a pen on top of that whiteout. In my experience, pencils don't stick to whiteout very well. And it's so frustrating when I have like the most important part of an exam question scanned in and you can see that there's something written. It's like the shadow of an answer, but it's over a whiteout and you just can't read it. It's so heartbreaking. Pen on whiteout. Don't write on the back of the page. Don't do it. The back of the page doesn't get scanned in. If there's not enough space to answer a question, write on the margins or on a different page. And if you're going to be writing on a different page, tell us what page it's on at the question. So you say further answers on page such and such under question such and such. And then make sure there's like a line between it so there's space. Don't write on the back of the page. Remember, humans are grading this. Read over your answers and edit for clarity. Put a box around the important parts of your answer. Don't write in paragraphs unless it's specifically asked for. If you can write the answer in bullet points, please do that. I have to read this. So many of these. I'm scanning for the correct answer. Make it easy for me to find. Arrows, stars. Don't panic. Be gentle with yourself. You're doing your best and it will be over soon. You are in a third-year genetics course. Objectively, you are smart. Look forward, not back. We're here in this moment. Let's do the best we can. Hack the panic cycle. If you feel lightheaded, breathe in for 5 seconds, hold your breath for 5 seconds, and then breathe out. Part of what makes people panic is a lack of oxygen, which happens when you hyperventilate. So by slowing your breathing, you can calm yourself down. Close your eyes. If you're feeling, sometimes you're panicking because you're overstimulated. Close your eyes and think of just one concept. The flavor of chocolate, a waterfall, the love that you have for your pet. Just one concept, just for a moment. Just be like, okay, this one thing is the only thing in the world that exists. And then we can come back. And remember, this team, we are in your corner. If a question is confusing, raise your hand. If you're panicking and you can't breathe, raise your hand. I'm actually trained for this. I can talk down panic attacks in like 30 seconds under my breath. I'm here for you. If you aren't sure if your pencil is dark enough, raise your hand. If you need a calculator, raise your hand. We are here for you. Look at that timing. Look, it's been almost exactly 50 minutes. Amazing. See, if only I could have done that for the lecture, for the guest lecture.
Practice is perfect.
It's true. I'm much more practiced at this thing. Okay, well, that is my pep talk and review and prep presentation for the Bisc302W final exam. And I believe in you. You can do the thing. Okay. Bye. Oh, how do I end the recording?